Right. Hopefully everyone can see the full screen mode. So yeah, today I'll just be talking about a little bit about my work, what I do. So the title of my talk today is Stomach corals, Sea Ice, and Antarctica, and how they all interlinked, and how we're using this novel new proxy as a way to understand Antarctica better. So I'd like to say just a quick thank you to my three supervisors, Erin McClymont and Mike Bentley from Durham, and Dominic Hodgson from the British Antarctic Survey. So without further ado, so just a little bit of outline of my talk today. Just going to introduce what is sea ice, why is it important. I've spent the morning talking to kids about sea ice, glaciers, so hopefully kind of switch back into an adult type brain, and it's not going to be too simple. And then my next point, I'm going to talk about what are snow petrels. So this is kind of the exciting, novel, new biological archive I'm going to be talking about, and where exactly I'm going to be, where these stomach coral deposits that I work on are from, and why that is important, what kind of methods and materials I use. So Olivia, she touched on earlier, I use a variety of different geochemical analytical methods to kind of analyze these stomach coral deposits and what kind of results I'm looking at and what this kind of means in terms of Antarctica in a broader sense, both in the past and also the future, because obviously we are a paleoclimate society. So sea ice, the facts and the gaps. So just a brief rundown of the facts. Hopefully these aren't too simple, and some of you might know a few of these facts. So the first one, there are many different species present in the Southern Ocean, including the snow petrel. So the snow petrel is endemic to Antarctica, and one of the many species present that we use to kind of think about how we can use them as a new biological archive. And unfortunately, as we all know, that sea ice has been declining, especially in the Weddell Sea area, which is actually my study area, until about 2006. But actually, since then, the sea ice thickness and variability has actually kind of remained quite stable and has actually increased, but then has since declined, obviously because of climate change and things like that. Hopefully I can minimize this here. Obviously, it's very large, surrounding all of Antarctica, and varies. And I think the key thing at the moment is that we have lots of different models from the past about how the kind of summer sea ice varies, and that's kind of what I'm going to be talking about in a few slides' time. So the Southern Ocean, obviously globally important, is home to quite a few of the big currents that kind of connect the world's global oceans. For example, the Antarctic circumpolar current and the Antarctic bottom waters, especially in the Weddell Sea, are really important for the transport of heat, a carbon sink, and also provide a habitat for the variety of prey that these snow petrels that I work on forage on. So the gaps, what is my project about? Why is there a need for a new proxy? So my study area, hopefully you can see, is the Weddell Sea area all around the Birkenhead Island and the Theron Mountains that I'll go into a bit later. And essentially it's a very inaccessible area covered in very thick multi-year sea ice. And so it means that when obviously us as scientists try and get to these areas, it's very inaccessible. And for that reason, as you can see on this map of Antarctica here, there are very few kind of ice cores, marine sediment cores and ice cores, and there aren't really very many that are very suitable around the Weddell Sea area. And for that reason, when we try and model a kind of summer sea ice extent or winter sea ice extent, these model outputs that we look at can vary massively. And that is a massive problem in terms of thinking about what's going to happen in the future, and also knowing what's happened in the past and how we can kind of connect sea ice to the ecosystems around us. I think it's pretty essential that we have these model outputs that agree with each other. And my kind of penultimate point is that these processes that influence sea ice melt, especially in the late Holocene. So a lot of stuff's been done the last ice age, the kind of last glacial maximum around 20,000 years ago. But I think the key thing at the moment that I'm looking at is the late Holocene. And there really isn't much like a great understanding of what drives sea ice melt, sea ice decline and sea ice increasing in the late Holocene, especially around my area. So I think the point is that we need this new proxy. We need a new proxy, and thankfully there is a new proxy that we can use. So if I go on, so getting to the main part of my talk, this new proxy, these are snow petrel stomach hole deposits. So the next few slides, I'll just talk about what they are, why are they important, why do we need to use them for the future of sea ice modelling and understanding climate change better. So this is a cartoon of a snow petrel. As I said earlier, they are endemic to Antarctica. They kind of look a bit like a pigeon. Well, they're kind of the size of a pigeon, size of a dove. They are pure white to camouflage in with the ice and the snow. And they have kind of black beaks and feet. Their typical prey will tend to be fish, krill and squid. So the krill will tend to be found further out in the kind of deeper portions of the ocean, whereas the fish will tend to be found close to the shoreline. And the squid, again, will tend to be found further out. So they're the kind of main components of krill, and that's what we're looking at in terms of their diet that I'll talk about a bit later on. So they'll typically have one chick breeding season. So there'll be breeding pairs. They'll kind of colonies will come back and forth to the same nesting environment. And when they're kind of in that post brood, they've had their chick, they're going out to feed and hunt. They will typically forage up to 500 kilometers in a single trip. So that would be a very, very long way, and is actually quite interesting and can tell us a lot about sea ice, kind of both at the shoreline, at the sea ice edge, and also 500 kilometers further north or further east or west, wherever they're going. So their stomach oils, so they have a very, very unusual habit. Similarly to llamas and I think a few other animals or mammals and maybe a few other birds, they will voluntarily regurgitate this thick, gooey stomach oil that's bright orange, it's a bit like bile. And they will typically do this as a defensive mechanism. So there's a bit of debate at the moment as to whether these are kind of more adults are doing this or whether they're chicks in the nest. Is this defensive mechanism against skuas. Skuas, albatrosses, those kind of larger birds that will typically prey on these nesting chicks or these nesting adults. Or there's a debate whether they could be a food source. So these stomach oils will contain partially digested, regurgitated prey remains. So whether that's fish, whether that's krill, or whether that's squid, that will be contained in this oil. So it'll be a very, very fatty, like protein filled kind of source from their stomach, basically. And it's whether that could be a food source as well, which might be a bit interesting. I'm not sure whether the chicks would like it. And yeah, as I said, is it the chick? Could it be the chick that's kind of regurgitating this stomach oil a bit more? And what's interesting is that they do sometimes spit at each other. But obviously that can compromise their kind of waterproofing, so they will tend to roll across the snow to get rid of the oil, which is very cute. So bringing kind of back to my project, so as Olivia said, my aim is to reconstruct snow petrel diet using these regurgitated stomach oil deposits, and from there reconstruct or attempt to reconstruct summer sea ice extent from my region, which is the Theron Mountains, which is the eastern side of the Filchner ice shelf of Antarctica. So I've got four main objectives to my project. The main aim is to kind of date these stomach oil deposits and look at the different stratigraphic integrity, so looking at the layers that are present, so which you'll see a bit later on. And then I'm using a mixture of kind of visual and geochemical analysis to characterize changes in stomach oil deposits through time. So looking at how this diet changes through time. And what's quite interesting is that because when they regurgitate the stomach oil deposit, it will build up outside of their nest, accumulate throughout time, so therefore gives quite a nice kind of age depth model and diet throughout time. And then use the results from the first two objectives to reconstruct snow petrel diet and assess how these changes in diet, whether they are eating a diet more rich in fish or krill, will it kind of coincides with sea ice scenarios in this foraging range of the Theron Mountains. So my study area, as I said, is the Theron Mountains in the Weddell Sea region. It's the Weddell Sea as outlined here on this map of Antarctica. is the largest Antarctic sea. It's very important. You've got the Weddell Gyre, which causes rotation of very cold, low salinity surface water northwards, so contributes to the wider global ocean circulation. And that's the same with the Antarctic bottom water, drawing cold, saline water all around Antarctica and further north. It's also very important because it's the site of the transient Weddell Sea polynya, which opens up every now and again and could be another potential food source for our snow petrels. So if I move further on, so the Theron Mountains. So my deposits were collected in 2015 by two of my supervisors, Mike and Dom. So that's labelled here, this nice pin here. So these are the Theron Mountains, which I think are slightly north of the Shackleton Range. The main geology is sandstone, siltstones, with some shales and mudstones. So sometimes in these fossilized stomach oil deposits, you'll see little pieces of bedrock within, which could be another potential kind of dating technique. And so this is a site of historical snow petrel colony. So you can see these three photos below. These are photos that my two supervisors took of the Theron Mountains. So the first one, you can see the Theron Mountains in the background. And what's interesting is that even though this is kind of a rock here, could be some sandstone, the stomach oil deposits are kind of draped across, and they're almost quite camouflaged around the rock. And so this middle photo is an example of a snow petrel with a little bit of fresh stomach oil here. So yeah, typically they will kind of regurgitate it just outside their nest. You can see how over time this will kind of change color to a kind of quite browny, dark orange type color and build up outside of their nests. And then on this far right photo, you've got the surrounding bedrock of, I think that's mudstone and some shales. So this is obviously quite high up, so a snow petrel will nest in these little crevices. And then this kind of lighter brown color would be the stomach oil deposit. So going on to my research and to the methods and how are we taking this stomach oil deposit and how are we making something kind of tangible and understandable out of it. So the process, we start off with taking a core. So these vary in size, you'll see in the next few photos. These vary in size from a few centimeters thick and long to I think over 30-40 centimeters long. So what we do is we take a core and take it firstly through the core scanner to do some XRF analysis, so X-ray fluorescence analysis, which will look at the main elements present in the stomach oil deposits. And then secondly, we will do some stable isotope measurements. So we will look at the carbon and nitrogen, total organic carbon and nitrogen content, and the two different stable isotopes of carbon and nitrogen as well. And then finally, we'll do some, I've been doing some fatty acid analysis, which I'll run through a bit more in a minute. So hopefully you can see this. So again, in a little bit more detail, so as I said, what does X-ray fluorescence, what does XRF tell us in terms of these deposits? So there are a few different elements that can tell us a bit about the diet. One of which, hopefully you can see, is the element copper. So there's been a few literature studies that have said that if you're going to see copper, you're going to see an increase, a spike in copper that's potentially linked to an increase in krill, which is found in their blood, so that we've been using that as a proxy. And then secondly, looking at stable isotopes. So carbon will tell us about the wider climatic trends at the time, so delta 13C. So we'll see different shifts in those. And delta 15N will tell us about trophic level. So the lower the delta 15N value could be indicative of more of a krill-based diet. Whereas as we move up the trophic levels, you'll see, we're hoping to see an increase in delta 15N. And so finally, the kind of key thing that we've been looking at are FAMEs, are fatty acids and fatty alcohols. So again, there's been in previous literature that's kind of worked on not just stomach oil deposits, but just kind of bird diet in general, have kind of given us these different proxies that we can use and to understand this better. So we've been using these three different saturated fatty acids here. So carbon 14, 16, 18 as an indicator for krill. and just quite kind of widely known. And that's kind of indicative of potential foraging more in a pelagic environment, so open ocean. It's going to be plentiful krill there to snack on. So secondly, we've got the carbon 18 and carbon 18.1, so isomer, the first isomer, is potentially more indicative of more of a fish-based diet, which people have kind of judged to be perhaps more foraging from more of a neritic environment, so close to the continental shelf, when the summer sea ice might have been further back towards the shelf, so it meant that these snow petrels wouldn't have to have flown and foraged so far, because why would they have flown, you know, all the way to the open ocean to have some krill when they could just forage along the sea ice edge? And finally, though kind of debated, carbon 16 is also could be a copepod signal, could be a squid signal as well. So that's kind of what we've been using as our basis for understanding a bit more about these stomach hole deposits. So polynyas. So polynyas, as I mentioned a bit earlier, like the Weddell Sea polynya, could heavily influence snow petrel foraging habits. So depending where they are, if the sea ice was really, really extensive, the snow petrels couldn't fly to the sea ice edge. So what would they do? Well, hopefully a polynya would open up here somewhere within the sea ice pack. So within these kind of mid-ocean polynyas, and then hopefully they would forage in these kind of areas, and these would be areas of kind of, you know, upwelling, productivity, and hopefully they'd find something to eat there. Also, where you've got the sea ice edge, it's very, very windy. You've got these kind of really strong katabatic winds flowing down, pushing the sea ice pack apart, and these could be potential areas for foraging. So just quickly have a look through these photos. So first of all, we take a core. So the first line of photos are essentially what we get given in the freezer. Very, very interesting looking. My ones at least are, you know, very rock filled, which, yeah, you can see pieces of granite or slate or shale, and they form these nice layers like this. And so what we do when we take a core, we are looking for these nice layers. So if we decide a core is suitable to continue work on, we want to see these really, really nice layers. So these are quite pale in colour, but I've got ones that the older the deposit, they'll be quite a lot darker in colour and might be very soily and interesting texture compared to these fresher ones. So this deposit in particular dates from, I think it was 2,000 years ago to around present day. So it's a very, very young deposit in comparison. So moving a bit further on, these are some examples of some Holocene deposits. So you've got these really, really nice layers, kind of dark yellow in colour. We've got some inclusions. These kind of black spots could be guano from the snow petrels themselves, but that's kind of a bit of a debate. They've not found to kind of change geochemistry too much, but it's just interesting. Some deposits have different amounts of guano in them. So you can see in the middle, we've got a core. So this will essentially go through the core scanner, and then we'll get some idea of the main, the total elemental compositions. So you can see on the far right, these are some examples of my pre-LGM deposits. So these date from around 40, 50,000 years ago to even older, which is pretty insane that they've managed to last this long. And I think the cold environment has really preserved them quite nicely. And you can see they really are quite different in terms of texture, in terms of color to the earlier deposits. So often when we take a core, we can kind of see that they're not really, they're quite swirly, they're quite mixed up, they're quite degraded. So unfortunately, probably about 5 or so of my much older deposits aren't very useful. So stable isotopes. So what we do when we have our core, we take one side of the core for our scanning, and then we take another side for some stable isotope measurements. So on the far left, you can see them all crammed into the freezer. I think we've taken over most of the Durham University cold store freezer department with all of these stomach hole deposits, with more to come. So you can see in the middle, this is kind of my setup for subsampling for stable isotopes. And on the far right, the way we kind of been doing it is we've been taking these kind of 2-3 millimeter biopsy hole punches straight down the core, so we can get a nice kind of idea throughout time of what diet and what the stabilized isotopes were doing. So onto the geochemical analysis. So essentially, same thing again, we subsample. So this far line of holes on the right would be our kind of subsampling for our fatty acids and our fatty alcohols. So basically the method is we do some total lipid extraction, which ends up with this kind of interesting looking liquid like this. And then the next day or the next few days, we'll do saponification and methylation, basically adding a methyl group to allow us to get some the fatty acid methyl esters, which we then run on the GCMS, try and identify and see what's going on. So my research, some key results. Hopefully I'll try and keep this simple, but yeah, feel free to pop in a question afterwards and I can answer it at the end. So these are the results from some of my radiocarbon analysis from dating these deposits. So what you can see, so I had about, I think it was 26 in total. So dating these places these quite clearly into two separate groups. So the large majority, we've got age on the bottom axis and altitude on the y-axis. You see quite a lot of these deposits do date to around 5,000–6,000 years ago to present. So kind of late Holocene, which is good because that was one of the questions that I was kind of asking, what's happening in the late Holocene in terms of summer sea ice extent in the Theron Mountains. There's this kind of questionable deposit in the middle, which we thought, not sure if that kind of makes sense. If the last glacial maximum was kind of 20,000 years ago, we're not really too sure. So we actually redated it and it placed it a lot later. So these deposits on the far right, these green ones here, are over 50,000 years ago. Years old, sorry. And so they really are very old. And the first thing we kind of looked at when we saw this, we thought, why is there a big gap in the middle? What could be the reason? Aside from, a potentially inhospitable environment during the last glacial maximum, what does this mean? So that's kind of what we were finding out. So this kind of, this here shows a few of the deposits I've been working on in terms of age. So at the top, we've got kind of very boring lab code to ID the deposits. And further down, these are photos of my deposits, kind of squished in or stretched depending on age. So a lot of them here are kind of, about 2,000 years. So that's kind of very late Holocene. And we've got this very nice deposit 130114 that stretches until about 5,000 years ago. But as you can see, it's very, very swirly. There's not, not many good layers to kind of work on. So unfortunately, we had to cull that from the group. And so this far right deposit here, this is one of our old ones that I showed earlier. So there is some layering there, but it looks completely different in terms of color. These are kind of these Holocene deposits are a lot brighter and orangier and sticky. So when we work on them, we have to take them out of the freezer quite quickly, otherwise they will probably melt everywhere. So this is kind of some of the data from my stable isotopes. So we've got the delta 15N on the y-axis, and we've got delta 13C on the x-axis. So kind of very similar to trends that have kind of looked at these deposits in the past. We've kind of got all of these Holocene deposits clumped together, kind of in the kind of more negative delta 13C. Whereas the kind of older, the pre-LGM deposits kind of slightly cluster in their own group, which is quite interesting. And some of them do range in delta 15N values. We've got a few anomalies. Not quite sure yet why. That could be to do with more guano in a deposit, meaning that there's some more nitrogen in the system. Or we're not too sure yet. There could be some rocks there that's kind of influencing the nitrogen values. So here, I know it's a lot of graphs, but I'll try and kind of talk through it kind of quite clearly. So these are the results from my XRF data for the Holocene. So what I've done, these are graphs of some copper data. So my aim has been to look at how has copper changed throughout time, and could copper be a proxy for krill. So what's interesting is that we can see that there are a lot of things going on. There's a lot of trends, not necessarily matching, but that's okay. We can definitely see that there's a lot of Holocene variability. So the time is on the y-axis. So that goes from about 2,200 years ago to about 170 years ago. And yeah, as you can see, there's a lot of variability going on. And I've kind of pinpointed some perhaps key areas where there might have been more of a krill-based diet. So they might have been, they might have been foraging further out into the open ocean. So then we can infer that the sea ice might have been either kind of further forwards, they're being forced out to the open ocean, and there are areas kind of where there might have been more of a fish-based diet, further closer to the sea ice edge and close to the shoreline, close to the continental shelf. So again, kind of a very busy graph. So these are kind of some of my fatty acid trends from the Holocene for two of my deposits. So for the first row, this is this first set of graphs. This is deposit 130111. So it dates from about 2,000 years ago to about 140 years ago. Got really nice layering, and some interesting things happening. So here I've got the main saturated fatty acids. So I've got carbon 14, 16, 18, and then I've got 16.1, which could be a potential proxy for degradation. So the kind of the lower value it is, more degradation there could be. And then I've got carbon 18.1, which could be an indicator for fish even. And then these last two graphs on the right, we've got delta 13C and delta 15N. So I think the main kind of key is that even though there's a lot going on, I think the longer the deposit, in which case this is, it's quite a long deposit, it definitely shows some overall trends. So even though we can't perhaps say anything directly about the diet, based on these kind of single points, we've definitely got cycles going on. So we've got cycles perhaps of stable climate, stable diet here, and then these peaks could indicate perhaps a slight change, whether or not it's due to polynyia or not. This could be an indicator of perhaps more of a krill-based diet, and then it kind of goes a bit more stable again. And there's something a bit crazy happening at the bottom, whether or not it's due to this rock here that I tried to avoid, but this could be a potential big shift in diet that we're kind of seeing mirroring across the stable isotopes as well. And down at the bottom, we've got another Holocene deposit that dates from around 2,000 years ago as well, to about 800 years ago. And I've taken some dates in between just to see if there's any differences in accumulation rate. The accumulation rate really varies. Obviously, it's not a sediment. It could be, 2 millimeters every 1,000 years, 2 millimeters, every regurgitation. So it really does depend. And this kind of latter deposit does show the same sorts of shifts that you get, periods of stable kind of dietary trends, and then some more crazy things happening, perhaps a big shift in diet that kind of corresponds to something happening in the stable isotopes as well. What's interesting about this deposit here, the nitrogen values really jump up quite massively, and we're still kind of yet to find a reason. Could be to do with in this deposit at the base, there are quite a few more rocks and inclusions of bedrock present, and that could impact the nitrogen values. But here it's quite interesting because they do jump up quite a few trophic levels that could perhaps not be to do with diet at all. So moving on, so these are some of my pre-LGM deposit results. So this was quite a nice one. So the one at the top, they've got some really nice layering. Unfortunately, we don't have a very good chronology on this because obviously it's beyond calibration, but it shows some pretty crazy things. I think it definitely shows that In each of these graphs, there's quite a high level of variability going on. So potentially it was a really, unstable period, and there were lots of shifts going on. And overall, you can kind of see that with the delta 13C and the delta 15N, they show kind of gradual decreases. Whereas in comparison, this deposit right at the bottom, it's a lot older. It does have layers, but it was very soily in texture. And I think, in all honesty, I think a lot of the fatty acids have degraded because there's really not a high amount of, the concentration is really quite low compared to the Holocene deposits, and we really don't see much to talk about. So I think it's an impact of degradation, but it could also be a very stable climate. So we're just not really seeing too much. But I think these deposits, because they vary, where they're left, whether they're outside the nest or they're inside a wind scoop, this wind scoop could really either be protecting the deposit, or when it's exposed to the open air, you can quite a lot of degradation can occur. So moving on. So again, looking at the different fatty acids in a different way. This is a Holocene deposit again. And these graphs basically show kind of box plots that basically show differences in concentration, hopefully show it in a bit of a better way. So the first line of graphs, we've got some of the saturated fatty acids. We've got carbon 13, carbon 14, 15 and 16. There is some debate whether carbon 15 could be a temperature proxy. We're not too sure yet. But I've kind of basically highlighted here that the concentrations here are really quite large, especially with carbon 16. And because of that, these kind of quite high concentrations of carbon 16, that's quite a good indicator of krill. Whereas you can see here carbon 18.1 and carbon 18, even though carbon 18 fatty acid could be an indicator of either krill or fish. In that case, we'd be looking at carbon 18.1, the isomer 1, to see what's happening with the fish. And you see it's kind of, the values do vary, but on the whole, they're a lot lower in terms of the saturated, compared to the saturated fatty acids. So here, this is looking at pre-LGM, so an older deposit, looking at the same thing, so comparing the concentrations, comparing the average values of concentrations of the saturated versus the unsaturated fatty acids. You can kind of see that the concentrations on the whole are a lot lower. Whether that's due to degradation or not, we're not too sure. Perhaps because the geochemistry is slightly different, that could indicate perhaps what we're seeing here in this pre-LGM deposit. It's not kind of mirroring the same environmental conditions that the Holocene deposit is kind of showing. So perhaps it could be, within a stadial period rather than interstadial period. So just to kind of run through this, because I'm aware of the time. So these graphs here, just again, are kind of looking at the different concentrations of the fatty acids. So just draw your attention to this first graph on the left. This is a graph of all my Holocene deposits and looking at the different concentrations of carbon 16, which is the krill indicator. So you can see here they do kind of vary in concentrations, but overall compared to the pre-LGM, which is this graph below, a lot of the concentrations in the Holocene deposits are a lot higher. So that could either indicate it's more of, on the whole, a high proportion of krill in their diet in the Holocene compared to pre-LGM, or it's a mixture of degradation as well. So you can see this bottom graph, the concentrations on the whole are a lot lower, so more in the region of 2,000 compared to perhaps 6,000 in the Holocene. Whereas what was interesting, I found that this green bar here, which is deposit 13018 from the pre-LGM, it has similar carbon 16 values to the Holocene value. So perhaps this could be, if we were to kind of estimate a date for this deposit, it could be within a similar environmental condition that the Holocene deposits show. In the middle, I've got a comparison of different ratios. So I've looked at, in the top graph, I've looked at comparing carbon-16 versus carbon-16-1 ratio, looking at the Holocene and the pre-LGM. So you can see that there's kind of quite a wider range in the Holocene, which I guess is to be expected. They're going to preserve, these fatty acids will degrade less, and there's going to be a wider range compared to the pre-LGM, which show slightly lower average and kind of just less of a range as well. So either these could be two different environments going on, or again, it's just a product of being a lot older. What's interesting though is that this bottom graph here shows a ratio of carbon-18 versus unsaturated 18-1 fatty acid, comparing the Holocene and the pre-LGM. So this is kind of a lot lower ratio in the Holocene compared to the pre-LGM. So the pre-LGM, perhaps that could have been more of a fish-based diet. If carbon-18, it could be an indicator of fish. So again, it's just something that I've been looking at, been playing at. What are these differences? What do they mean? Can I read a pattern into it or not? Not too sure yet. And then these last two graphs, we're looking at the the carbon-18 to 18-1 ratio, comparing Holocene. And this top graph to pre-LGM. What was interesting is that the pre-LGM ratios were a lot higher and more consistent in terms of range. Whereas the Holocene deposits, the range was a lot lower. Was a lot lower. Whereas this deposit here, 130113, the range was kind of quite a lot higher actually. So yeah, it was very interesting just to kind of compare, look at perhaps this was less of a fish-based diet here, which would kind of match what was happening with this first graph. Maybe there's more krill in this kind of set of deposits here, because these set of Holocene deposits all range from about 2,000 years ago to present. So kind of just look at my last few graphs. So these are two pie charts that demonstrate the Holocene versus the pre-LGM fatty acid trends. So on the whole, at first glance, you kind of see that perhaps for a lot of these fatty acids, they were very, very kind of similar across, from the Holocene to the pre-LGM. I think the big caveat to say, obviously, with the Holocene deposits, we have exact ages, we kind of know their chronology, whereas the pre-LGM deposits, we have no idea how old they are. My deposits in particular tend to, their time span seems to be between 1,000 to 2,000 years. So if they are, say, 50,000 years old, the oldest, the base of the deposit is probably going to be 51,000 years, and the top would be 50,000 years. So it is quite interesting, kind of looking at the time span and looking at how the geochemistry changes. And I think on the whole, even though this geochemistry on first glance can look quite similar, we can kind of see a slight increase in kind of average concentration for the carbon-16-1 fatty acids, which not too sure what that means yet. I think because I was expecting degradation to perhaps lower the average concentration, but perhaps it's a dietary indicator, or perhaps it's coming from somewhere else. We're not too sure what's driving it yet. So I'm going to skip this slide in the interest of time, but it's basically looking at ice core data and how I can use it to try and justify and ground truth my fatty acid concentrations to see if I'm on the right lines in terms of chronology. So let me just check. Yeah, so I'll just go on to this slide now. So as I said, kind of looking at how can I kind of ground truth what I'm thinking. How I've got these dates, I've got these deposits, but how can I make sure that these dates are certain, because these dates obviously come with hundreds to 200 years uncertainty. So these graphs show, so the orange line shows my titanium data that I've got from my XRF data for each deposit. So I've got an example of 3 different deposits here. And the blue line is an ice core. So I've been looking at dust flux from an ice core, and I've been looking to see if my titanium data matches up with the dust flux data. And looking at this first graph, yes, it doesn't exactly match up, but you can see similar sorts of spikes happening in the right sort of places. So I can kind of think that maybe perhaps my chronology, perhaps my age constraint might be along the right lines. And you can see here on this top right graph, you kind of got a spike coming up at the same time as the dust flux, and the titanium again, doing the similar sorts of things to the dust flux. So that's kind of what I've been thinking of, what's best to kind of ground truth my data, and the same on this last graph. So just to kind of wrap up, hopefully I've not been too boring today, or too simple, or too complicated. Obviously it's quite a new proxy, so there's a lot going on, a lot of questions, and it's a bit unusual. So the findings, I've been finding, centennial scale variability in diet across Holocene. I think I've also been finding millennial scale. So I've definitely found periods of quite intense variability in signals across both my isotopes, my fatty acids, and I've also been looking at fatty alcohols, which has been interesting as well. So there's been periods that I found of quite stable diet, and then all of a sudden you've got massive spikes in my signals, which could perhaps indicate a shift in environment, whether the sea ice was moving further out, so snow petrels had to find a different source of food, so perhaps they had to find polynya, which I've been looking at to see maybe a polynya was present if my carbon-13 has been spiking, but my krill has been going down. Perhaps that could be a polynya. And again, the main thing that came out as well, we've got this late occupation of snow petrels in the Theron Mountains. So my deposits, as I said earlier, clump into two different groups. The Holocene set clump from about 6,000 years ago to present. But current models of deglaciation in the area put that more around, sea ice should have started to melt, and the ice sheet should have started to kind of draw back around 8 to 10,000 years ago. So why is there that late occupation? Perhaps there was just simply too inhospitable until a polynya opened up, in which case that's perhaps what I'm finding in these dates. And again, we've seen this increase of C16.1 in my pre-LGM deposits, so perhaps the geochemistry being a bit different. Why is that? Perhaps my pre-LGM deposits could indicate a completely different environment to what we were seeing in the Holocene deposits, and could help kind of better constrain our chronology of what's happening in the area. And again, perhaps the use of titanium data alongside ice cores could be used to reduce this chronology, which is a key factor at the moment. But I think I've kind of hopefully shown that there are some kind of different multi-proxy approaches that can kind of be used to investigate our stomachal deposits. And though it is a bit up in the air, I think it's really exciting what can happen, what we can use these for. And it's been great because there's been quite a lot of stomachal deposits collected from all around the coast of Antarctica, so we can kind of draw a nice big picture of what's happening to sea ice, kind of way back in time to quite recently. So what is next? It's my last slide, you'd be pleased to know. So future work. So we have some other members from the team going out to collect some more deposits, which is very great. We've got some people working on diatoms, because that can be a kind of a direct indicator of sea level. So that's taking some time, but hopefully we can use those in conjunction with our other proxies to kind of get a better indication of what's happening in the past and 2,000 years ago to present. More locations. So currently the stomachal deposits, I'm the kind of furthest west, I guess, in the Theron Mountains, and we've got some more deposits stretching along the coastline. We've got tracking data to complement that, so people that have in our group that have tracked these snow petrels, looked at their foraging range, so we can get some nice overlaps in terms of geochemistry. And it'll be really interesting to see if the geochemistry is similar along the coastline, if it's different, if the ages overlap, and if they overlap, that's great. What can our results tell us about the future? And especially in terms of modelling. So even though my kind of work isn't to do with modelling, as such, hopefully what we find out can, especially about sea ice variability, can tell us about, can hopefully feed into modelling data to kind of better constrain these disagreements and disagreements in model outcomes and summer sea ice variability in Antarctica, and hopefully work towards some kind of picture of how, if we know what happened to sea ice in the past, how might it change in the future. So yeah, I think that's everything I had. Thank you for listening. I see there's been a few questions, but yeah, thank you so much, and hopefully it made sense. Yeah, thank you very much.